All right, so I think the recording has started, should be recording right now. All right, fantastic. So what we would like to do today is just to go over the paradigm and go over this fantastic initiative, which I call it the biggest brain stimulation experiment that's ever been done. And we hope that the presentation wouldn't take up too much of time, hopefully maybe at a maximum 20 minutes or so that we have lots of time for questions. So this meeting, I think, has been scheduled for 1 hour, but I have left another half hour after this meeting, so I could at least stick around for another half hour, right? If we need the time, but I do realize that some of you may have to leave after an hour sharp. Okay. So if it's okay, I will now start the presentation. I'm gonna share my screen. And get my slides up. So, okay, all right. Can you see the slides?
Yes.
Yes.
Good.
Yes.
And all right. Good. So yeah, thank you everyone for coming to this our official kickoff meeting for the tax challenge. Just to get everyone on the same board, and I realized that maybe for some of you this is repetition because you already know that. But I thought, nevertheless, it would be nice to start with giving a little bit of context. So this is an international multi-center initiative for the non-invasive brain stimulation community to address the question whether TSCS is actually effective in modulating behavioral performance. I guess many of you, certainly I, had my fair share of TSCS experiments that didn't work out in the lab, which is why we arrived at this point. And we don't know what the true effect size is of TACs. And so that's what we want to address in this multi-center initiative, which is basically what this point is about. So we want to do one big study, which is well designed and well powered. And how are we going to do that? So we will test for online effects of TSCS on behavior that result from the phasic current modulation. So in essence, we test the question whether, the TSCS phase that is presumably imposed on the brain, whether that affects perception behavior, whether it renders some phases more facilitatory for perception of a brief stimulus than other phases. We do already, within the review process, however, there were some questions about offline effects, and we did also include some hypotheses about potential offline effects, but our focus has always been and will be on the online effects of TSCS. Yes, our commitment is to promote the quality of non-invasive brain stimulation research through methodological rigor, acquisition of a huge data set, and scientific transparency and data sharing. Yeah, so everything we do should be hopefully transparent. At least that's what we aspire to be transparent, to share our data, and basically create this open this resource that is open to the whole community. So just as a reminder, that's the steering team which has been behind this initiative right from the start. So thanks everyone on the steering team for driving this forward. and for attending countless meetings and many discussions. Okay, so here we are. We now have 44 participating labs, which are all listed on this website. By the way, if you haven't made a note of this website, then please do, because this is one of the main sources of information. So if you go to the website, I'm gonna click on the link. It takes you here. So that's all the participating labs. So you can see we have 44 labs who will support this initiative with data collection. If you have already completed data collection, you get a green sticker like we did. We already completed data collection. So this will also serve us as a record of progress. So you can check in regularly to see who's completed the data collection already, which is also useful for things like device sharing, right? Which we come to later. And we also have a nice map of the world where all the participating labs are, which I think is pretty impressive, actually. Okay, let's go back to the presentation. So yeah, as I said, we will test for online effects of TSCS on behavior that result from that phasic current modulation. And we have designed a paradigm which should also control for sensory confounds. So most importantly, the stimulation of the retina and somatosensory stimulation or transcutaneous effects, as they are called. We will be recruiting 880 participants. So that's basically 20 participants per lab. And yeah, the outcomes of the experimental condition, which is the one where we stimulate the occipital cortex, will be contrasted with two thoroughly designed peripheral control conditions. So yeah, our objective is to rigorously examine the real-time impact of TSCS on behavior, to establish whether TSCS-induced cortical stimulation is effective in rhythmically modulating visual performance at the current intensities that are routinely used in humans. Okay, so here I'm giving a brief overview over the experimental procedure. We do have a shared protocol, so maybe the exact protocol might slightly differ from lab to lab, although the way in which the experiment is run should be the same across participants. And the Trento group, Carlo Minussi's lab, has kindly provided a template for a shared protocol, which you can download also on our GitHub folder. So just to make one important point right away, this GitHub repository, this is your one-stop for all the resources that you need for this experiment. So I will come back to that multiple times in the presentation. But essentially, this is where we have all the code for analyzing the data, all the code for organizing the data, and also all the other resources, like, for instance, the standard operating procedures, the questionnaires, and files for printing the 3D electrode holders. This is also where we have deposited our stage 1 registered report. So if you need to refer to our registered report, then please refer to it on this website. This is important because since the acceptance of our paper, after the acceptance, we realized there was a small error regarding the electrode positions, not the stimulation ones, but for the EEG recording or for the stimulation artifact recording, which we have clarified. So, right, so I can't scroll further down here, but you can download the registered, the correct version of the registered report on that website. So yeah, if you need to refer to it, please do that on that website. This is your one-stop for all the resources. And this is the shared protocol that you can also download here Okay, right. So each lab will collect 20 participants. And one lab session, we originally estimated that to be 2.5 hours, but after after we have ran this whole experiment, I think starting with an assumption that it's more around 3.5 hours might be more realistic. Maybe once you are very experienced, you might be able to get it down to 3 hours, but I don't think we could actually get it to run within 2.5 hours. So yeah, it takes a little bit longer than we originally anticipated. And then, yeah, you take the informed consent as a first step. And then there are some pre-stimulation questionnaires. Again, these questionnaires can be downloaded from our GitHub site. So there's in resources, you can see questionnaires. And there you see the questionnaire for the demographic information, exclusion criteria, the impedance check form, and we make a note of the impedance before and after the experiment, and also the TSCS sensations questionnaire. So this is where we record after the stimulation conditions, the sensations that the participants felt. And then you train the participant on the task. So you run a staircase where we titrate the intensity of the LED changes on the box. And this is all explained in SOP 3. So the SOPs you can download on the resources, SOPs. And yeah, SOP 3 that details the experimental procedure. Then you conduct the TSCS thresholding procedure. So this is where you estimate the intensity of the TSCS stimulus. I'm just seeing there's something in the chat. Oh yeah, here is the chat message. Okay. Okay, right. So in SOP 4, that's where you that explains how you do the thresholding. And again, you find this SOP on the SOPs on our GitHub page. Then you record the EEG pre-stimulation if you have the setup to do so. You may remember that one of the reviewers said we should control for individual alpha frequency. So that is why we record the EEG pre-stimulation, also post-stimulation. And I think most labs will be able to do that. There are 1 or 2 labs who can't do it, and that's okay. So, right. And then after you've collected the EEG data, you've recorded it, eyes open and eyes closed, then you do the behavioral task. And then after each block, and we have 13 blocks, you have the participant fill out the perceptual sensations questionnaire. Then we do the post-stimulation EEG. So after the 13 blocks have been conducted, you again record the EEG, eyes open, eyes closed. Most of you will do that. And I think, yeah, the 1 or 2 labs who can't do it, they won't do it, and that's fine. And then there's post-stimulation questionnaires, which is, I think, mainly the impedance check form. So in the impedance check will be done before the stimulation and after the stimulation. So if you download this form, you will see a section that applies to before and the section that applies to afterwards. And as a last step, when you've collected all the data, you will upload your data on OSF. And how you should do that is specified in SOP 5, which I actually have updated. So later this week, I will ask Florian or Ines to update the SOP 5 because we have generated some scripts that allow you to check your data before you upload it, whether it conforms with our requirements. So that, in a nutshell, is the experimental paradigm. As I said, your one-stop for all resources is this website. So please, please make a note of that website. That's where you find the SOPs and other useful things. And this is just a description of what each SOP explains. So SOP 1 explains how to build the TSCS cap. And there are also the files for downloading for the 3D printer to print the electrode holders. SOP 2 explains how to set up and use the TSCS challenge device. So that's the little that's the Arduino and the box with the LEDs and so on. SOP 3 explains the experimental procedure. And yeah, note that we made some very recent changes to the electrode placements. Which is basically, we had in the previous version, I think we said that we're gonna put one electrode on AF but that was wrong. We put the electrode on FP. And SOP 4 is about the TSCS stimulation intensity titration procedure. SOP 5 explains the data format and how to upload it on OSF. And SOP 6 is about counterbalancing. Yeah, and you also have a link to the questionnaires with these 4 different questionnaires. And yeah, as I said, the registered report is also on this GitHub, and also a template from the Trento group, which I found very useful. Yeah. We also have a folder in that same repository for analysis, and we have organized it in such a way that we have scripts for analyzing behavioral data. These are not yet complete. The analysis script for analyzing the rhythmic modulation of behavior, that I think is complete, but some other analysis scripts for the other hypothesis, they still need to be written, and Benedikt Zöfel is the person who is in charge of that. Thanks, Benedikt. We also have scripts for extracting the individual alpha frequency. So if you record the EEG, then you will also have to add the information of the individual alpha frequency into a file which is called metadata. And you can find the script for extracting the individual alpha frequency on that GitHub repository. Just note, because we have so far only recorded data using brain vision, this script so far is only suitable for brain vision, for reading brain vision data. So you may have to change the script for different data formats, but I hope that shouldn't be too tricky. And then we have another folder which is called Meta, where we have scripts for data sharing and data formatting. So essentially there you have 2 scripts. I'm gonna just quickly go to that folder. So there you have 2 scripts. One is called Bitsify, and this brings your EEG data into the bits format, which then conforms to our data sharing. Yeah, to the format that we need for sharing the data. And this is a script that's called file naming check, which makes sure that you have all your files complete and the names are correct. If files have unexpected names or incomplete, you get an error message, and then you can fix those and run the script until it says all data is okay, and then you can upload it. And we hope that this saves us some time later on, so that we can then just grab the data and essentially analyze it. Okay. We also have the Discord channel. So this is our one-stop for handling all communications. So previously we used a lot of emails, but we found that wasn't very efficient. And also only a few people had access to the email accounts. So that's why we switched over to Discord. So if you have questions, then please post them on Discord. We have many different channels that you can use. And I'm gonna just open the Discord page, which hopefully is somewhere here. No, it isn't, but I can just quickly go to Discord then. All right, so this is our Discord space. And I'm gonna post if you have already signed up, then that's great. And you can see we have all these different channels for general questions, technical issues, procedures, ethics, publication. The steering committee, I think, is only for the steering committee members, and analysis and device sharing. So we use this for communication. If you haven't signed up to Discord, then I will post the link at the end of the presentation, and you can click on the link and sign up. And please do, because this is what we use for communicating. Okay, I'm almost there. Just a few more slides, and then we can go into the questions. Yeah, I would also encourage you to use Discord also to share your progress or any setbacks or anything. I think we should use this to create a little bit of a community spirit, because an obvious challenge is that we're all living in different parts of the world. So in order to give us a sense of, we're in this together type stuff, I think we should also use Discord for that. Yeah, so some frequently asked questions that already came up deal with ethics and device sharing, and for both we have a specified channel on Discord. So yeah, so do use that. Okay, so last point. This is about the timeline. So I've actually added here when this whole thing started. So this whole project actually started an embarrassing, what is this? An embarrassing 7 years ago in January 2018, where Benedikt Zöfel organized a meeting in Cambridge where we came together and we conceived the idea of this tax challenge. So yeah, and then many discussions happened when we designed the experiment and piloted, and also COVID, of course. But now we're gonna hit, I think, the final stretch of this initiative. We have closed the call for participating labs. We have our stage 1 report submitted, revised, and accepted. So that was a big milestone, which was only possible because of you guys. So thanks for that. And yeah, so today we have our kickoff meeting, and we plan to have the data collection hopefully finished by the end of August 2026. All right, so that's it. Just as a reminder, this is the website for our tax challenge, where you can find also the links to the GitHub repository and all the other things. And this is the link to join the Discord channel. I'm going to just copy paste the link into the chat.
Simon, I've done it.
Oh, thanks. You've posted the. Okay, cool. So that's why I'm now getting lots of funny noises, because people joined the Discord. That's great. I'm just gonna mute this for now. Okay. All right. So yeah, that's me. That basically was the presentation. I'm now gonna stop screen sharing and let you ask any questions that you may have. Since there's so many of us here, may I ask you to raise a hand if you have a question, or type it into the chat. Daria.
Hi, thanks. Hi, everyone. Hi, Simon. Very exciting, actually, I don't know where to look. Nice to see you all. Simon, you know, maybe, or we have been in touch, or my doctoral student, who's also somewhere here on the third slide, I think, has been in touch with your PhDs, and that went wonderful. I think they clarified almost everything. So thanks for this, and for all your work also. And we are ready to start. Actually, she was on a separate doctoral semester. First question, practical, is you have three conditions or three orders and 20 participants. So that was one of the questions. Don't we need 21?
Yeah, that's thank you for asking the question. This is also something that occurred to us when we ran the experiment. And this is also why we then generated this SOP number 6.
Okay.
So the idea is that we stick to our 20 participants per lab, and every lab generates their own counterbalanced sequence
Okay.
of orders. And because that is randomized, it should basically even out. So we should have an imbalance, if that makes sense. So this should be, yeah. So look for SOP 6. If there is anything unclear, then let us know.
It's not online yet, right? Because I tried to download it.
Oh, it is. Oh, sorry. It is online.
Okay.
So if you go to
SOP 6, yeah. But anyway, I will manage this. And do we have to pay at any point attention to counterbalancing between males and females then, or is that also in there? So if it's in there, so don't answer, but yeah.
So we did not take that into account. Hopefully with 880 participants, it balances itself out. This is not something we have taken into account. And because the protocol has been accepted, I don't think we can make such changes now. So
we just run this SOP-based assignment, and then we are good.
Correct. I'm just going to share my screen so you can see this is it. So SOP number 6, double blinding, assigning sequences. Yeah. Yeah, and you managed to link it to download it, but yeah, I will manage this. I didn't so far, but yeah, I will. Another question is out of curiosity, because I have been discussing this with Hannah, my doctoral students, is the EMLA cream. We would be kind of happy to use this, and depending on what you prefer, but this just adds like logistically like an hour because of the absorption, and we have been discussing what to, but the questionnaires and everything is not like in between, it's just an additional hour. So what do you recommend or prefer? So I mean, maybe that's a question for the whole of the steering committee, but I'm happy to give you what I think. So I would prefer if people who can use the EMLA cream do use it, because I think more people will not be using it. And it just gives us an opportunity to explore what happens with higher stimulation intensities, because inevitably for the EMLA cream, we will have higher higher stimulation intensities. And I think there is, one reason. So one argument would be more gives more. Higher intensities is good. More current in the brain should have strong effects. But I do think there could also be an argument made that high intensities could have less of an effect based on literature that I've seen. So to cut the long story short, I think that if those labs who can use the EMLA cream do use it, because it gives us just this extra layer of this extra question that we can address. But of course, as you know, it adds probably another hour, maybe a little bit less, because whilst you wait for the EMLA cream to soak in, you can do other stuff with the participant and prepare them. But it does add time to the experiment, and also adds another layer of complexity getting it through ethics, which for some may be a problem. So actually we do have ethics. It was very, it was an argument, but I won kind of. It's through ethics already, but we have been discussing if we want to use this.
still, but yeah, I will talk to her. And then two small further things, and then I give time to the others as well. You mentioned this extra questions, and we discussed this as it is the doctoral thesis, obviously, and it had to have like content besides like giving the data to you, and we are lucky that we can use participants, like re-invite them who already have MR structural brain images, or we will scan them as well. And Agnes is a fan, as you know, of sleep and the whole chronotype thing, so we added also 2 questionnaires, and the question is basically if any lab would want to join this, to have like a sub question, like a secondary question with individualized computational modeling and sleep, so we can pool like then data together.
Okay, sounds fantastic.
So this will be just, we will have it for our 20 subjects, and we can share, of course, these 2 sleep and chronotype questionnaires.
Okay.
And if people have access to MR, we only do this anatomical T1, T2, and would like to, then we would have like a sub study.
Yeah, I think this sounds great. I guess you can use the Discord channel to just
sure.
make a shout out and invite everyone who wants to join. So
Perfect.
I have 2 other people asking questions. One is called Abteilung Allgemeinmedizin.
Yes, I'm sorry.
That's
I'm
Hannah.
logged in
That's-
Yes, I'm Hannah. I'm the doctoral student of Daria. I'm logged in a weird
Okay.
Zoom account. Yeah. I had one additional question to the EMLA cream, because while talking to other people in other lab who also not do TACS stimulation but TDCS, they've experienced that it's enough to let the cream sink in for 20 minutes. So I was wondering whether it would be an idea to change the protocol for the EMLA cream to maybe an entire duration of less than an hour, whether that would maybe make sense, because that would make the logistical effort of it a bit smaller. But I mean, if that's not an option, then we can also stick to the 1 hour. But we just noticed while piloting that it is just a lot of additional time, and the experiment is long anyway. Yes.
Maybe Ole, Till, sorry. Not Ole Jensen, Till Ole Bergman. Till, do you want to answer that?
Yeah, so hi everyone. I also had a question like to ourselves on the list regarding EMLA cream, because we're also planning to use it. Ethics are still like pending, but I have no doubts that we will be able to. And so I think we should use it exactly according to like the guidelines that come with the cream, not deviate from that, because otherwise we will have always like the doubt that possibly it wasn't enough, right? Because we didn't use it as it should have been used. I know it's adding an hour, and I think we should discuss maybe having a look at the presentation again with the pipeline, where to edit, and whether it's actually possible to edit somewhere that we can make use of the time in between, or whether we need to edit at some early time point before we go into the entire training and titration procedures, etc. And I think it's possible that we have to actually add this 1 hour, which, well, is maybe another, depending on your local standards, 10 or 15 euro payment, but might be worth it, and might be most clean if you not try to like interleave it with the other procedures too much, because you have to clean it up afterwards and dry it again, right, before you can actually get the electrodes in position, and you want to have the electrodes in position when you do all the titrations, etc.
Yeah.
So I think it has to happen relatively early in the procedures.
I think so. I'm just looking at the SOP. SOP number 3 is what explains how to add the EMLA cream, and it is important information that these SOPs are part of our pre-registered report. So I don't think that we can deviate from anything critically from that, and we do say 60 minutes in that SOP. So I do think we have
But
to wait 2 minutes.
we just don't specify when exactly, right? It is applied. So that is a small shortcoming, I think, of the SOP.
Yeah.
Oh,
Yeah.
what do
Yeah,
we?
but
We
no,
do yes, sorry.
we,
Yeah,
Wait,
so
please.
we do. So I just say I would fully agree that I don't think we should deviate from this because, and for everyone, the SOPs they were written and they are part of the registered report. So deviating from this will be a problem, and will, and for our multi-site study, this will be an issue if people start deviating from the SOPs. And there is an order on SOP 3 for the EMLA cream when to apply before everything else is done. If something in the SOP is unclear in terms of like exactly the order and what you can do in between, we did realize that we needed kind of like a master protocol explaining when to apply each SOP. Let us know and we can go back and clarify that. So as a first thing, check the SOPs. If it's not entirely clear, let us know and we'll consult you guys and we'll make sure that everything is clear and the order is clear.
Yeah, I think you make a very good point, Ines, in that we can change the SOPs if anything is not clear or if there is a mistake, but we cannot change it regarding anything that affects the procedure. Giovanna?
Yeah, thanks. Hi, Giovanna from Belgrade. For those who don't know me, I have a question which is not about the EMLA cream. It's about the amount of light in the room where we are testing. How is that defined? How do we standardize it, or do we don't standardize it, or what do we do about that? It's an open question.
Yeah. So to the best of my knowledge is we somewhere in our SOP or in the shared protocol, I think we mentioned that the room should be dimly lighted. So it shouldn't be super bright, shouldn't be super dark. It should be somewhere in between. We do not specify that any further than that. I think we don't have like, it should have this and that many lumen.
Okay, so it's up to us to make it optimal within our conditions for participants to not to fall asleep, but also to see the luminous change.
Exactly. Yeah.
Okay.
So maybe just briefly, the idea behind this is, so we consider dark adaptation and everything, but this makes it more complicated. Then we need a certain amount of time to adapt to darkness, and then the experimenter would need to operate in darkness or with extra time during the
4
preparation.
hours is
So exactly. So this is why just dimly lit, and the rest is taken care of by the titration procedure.
Okay.
Right? So it will vary, so it's more ecologically valid. But yeah.
So I just wanted to be clear, clear about that. And one more thing that I had as a question was for the resting state EEG. The idea is to record from one electrode.
Correct.
Yeah.
So you have, we have 3 electrodes placed in the scalp. One is the reference, one is the ground, and one is the actual active electrode.
Yeah. So only one?
Yeah.
Okay.
Yeah.
No backup needed.
No.
Okay. Okay, thanks.
Great. Malte.
Yeah, thanks. I have one question regarding the ethics. So I handed in the ethics proposal 2 weeks ago, and then the ethics committee came back to me asking whether I would be the first center in Germany asking for ethics. Because if so, then the first center in Germany could become something like the German leader, and then all other departments in Germany don't have to do a full ethics proposal, but only what is often called Anzeige, so a short version of the ethics, which could reduce the workload. But I was not sure whether someone in Germany already has ethics and maybe someone is already the overall study leader in Germany. So maybe we can coordinate that a bit.
I think Daria can answer that.
Yeah, exactly. So I don't know about the others, but we are in Germany and we have ethics. And I know what you mentioned about this fact that it's like a Zweitvotum, like a second vote you can hand in. And if you want, we can share our ethics documents. We have written them from scratch because we didn't know if any German institution already had a vote, but we do. So if you benefit from it, we can share it.
Okay, that sounds
Hannah.
great. Yeah.
In Discord, right? Again, so we will share everything in Discord.
Okay.
Yeah, if then there's need, just to add, if then becomes something like an addition, Daria, as you proposed, and we want to centralize SOPs and things, we can add to the GitHub, but I would say let's start from in a more anarchic fashion with people suggesting, and then if we move on to something that needs to
yeah, you can also put it on GitHub.
Exactly, yeah.
I can just share the whole documents. I mean, it involves like a study protocol and this Teilnehmer info, so this information for the participants and so on. And because we have like MR scans and information to this, but that you can ignore obviously and say you don't do it if you don't, and then you can just share it. Sure.
There is a question by, I don't know how to pronounce the name. I'll give it a try. Castellano from BINICNAS CIBIT, Coimbra. Yeah, so he, I guess, is asking that it would be great if someone doing a real experiment could be live stream or record the trial for all the steps. I do think that, so I'm aware that my two people from my lab, Yifan and Chofan, who have done the experiment, they have been in touch with other labs and arranged for calls to help them. I think this could be something we can also organize via the Discord channel. So if someone is like collecting data from a participant, and then they could arrange a call with any labs who want to basically see the steps. I think we could do it like this.
Or if anyone wants to volunteer to create material and resources, they're also great, right? So if you're starting and if you want to record something, that would also be good.
Yeah. Okay, I'm just looking out for questions. Don't see any hands. Till?
Yeah. Quick one. I should know, but so we were planning for different alternative solutions for the montage switching, ranging from completely manual up to fully automated. I'm not sure whether we ever finalized the fully automated montage switching device.
No, we haven't. No. So the one thing that we do have is a device which switches the montages. Sorry, I want to find a picture of the device. Can't find it at the moment.
I can put one in the links. It's on project requirements and status, and then there is the montage switching, which is kind of an analog, so it has a dial.
Yes, correct.
And the link for it is on the chat. So I have one of those. I think Simon too, and other labs have too. So that's an option to make things faster.
Yep. So it's basically, I mean, you're probably all looking at it right now, but just in case you don't, this is what it looks like. Yeah, I think that we sent ours to a lab in China along with our other device because we finished data analysis. But yeah, so we use this device to switch between the montages. We have it on the towel so that the experimenter who interacts with the participant doesn't know which setup it is, because yeah, we want to have this double-blinded. Maybe also a point to reinforce here that we have our experiment double-blinded, so the person who's interacting with the participant should not know which stimulation condition is currently carried out. Yeah, we found this device works very well. And we've tested it. Like we measured the voltage that actually we get out of the device, and yeah, it works. Okay. Do we have any more questions? Can I? Till, yeah.
So just getting back to the male-female participant ratio. So I don't think it will average out across labs, right? Depending on the number of participating psychology labs, well, then most likely psychology students are going to participate as subjects, and then we'll have most likely way more female than male participants. Maybe that's not a problem at all, but I guess there's no way around this now, right? Unless we somehow keep track of the overall number of male and female participants and see whether some labs can make up for the imbalance that is created in other labs, like a little bit more like in the flow along data collection.
Till what I initially meant is we are normally trying to, I mean, except the psychology, we are trying to equalize at least 10-10, like in this case. And what I meant, if we keep, we try to also additionally keep track that both 10-10 have comparable counterbalancing. So not the 10 females start with, you know, a nodal and sham and so on. That's what I meant, but you're right, of course. But in our case, we would try to have as many females as males, for instance.
And actually, even if it's not, yeah, so that's a good point. And even if we don't have it in our SOPs for now, and we can't change this as a requirement, we could still encourage all the labs to try and get in a balanced sample, right?
At least like 12-8 or something. Yeah, I would not go for just random and invite all the first, like, yeah, volunteers who are females and probably 20 years old. And so, yeah, so this is, I would also go for more variability.
Yeah. Could I ask for a show of hands who will who already handed in their ethics? Could you just raise your hand on the reactions thing, if that's okay? So again, okay, so it's 1, 2, 3, 4, 5. Oh, okay, wow, okay, so that is actually, that's quite a lot of labs. Okay, good stuff. So that's great. And can I ask for who is planning to have their ethics sorted by the end of the year?
Is this exclusive with the previous question, like sorted in terms of handed in, or then having a positive vote?
It's inclusive with the previous question. So if you already have your ethics, you can raise your hand again. I just want to get a feel for which labs we can expect starting data collection in the new year, basically. Okay. All right. Good. So that's quite a lot. That's great. And can I ask the question, who of you will need to share it? So okay, so who will need to borrow a device for data collection? Okay, so that's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Okay, so that's about 12 labs. So right, so the device sharing, I think that this is something we need to sort out. So obviously those labs who have completed, if they want, they can share the device. And this is at my lab. We've already shipped our device to a lab in China, so they can start with data collection. And Eddie, you have a couple of devices, right, that are available.
So we have built 25 in total. We've sold 17 full devices, 2 kits. So that's 19 devices that belong to labs who have bought devices. And then of those 25, there are 6 that we've made that are all out on loan.
Okay.
So all the devices we've made are out there somewhere. So now it's a case of, because of the limited amount of those Teensy Duinos we can buy, because they've stopped producing them, it's pretty much a case now of labs will have to start sharing because we're not going to be able to produce many more.
Okay, but if we have 25 devices, well, 19 devices that are kicking about, and we have 6 devices that could be borrowed, basically, then I think this should be fine, right? That should see us through.
Yeah, I should think so. I think as long as people are pretty good at sharing them and updating when they've finished their data collection. We know where all the devices are, obviously, because we ship them. So we're keeping track of it. It's just we need to know when people finish so we know when we can send them on.
Okay, so right, so if you need to borrow a device and you haven't sourced the device already, then just as soon as you know when you start data collection, maybe 1 or 2 weeks before you actually start, get in touch via the Discord channel, device sharing, and then Eddie, you will monitor that, and then we can make sure you have the device when you need it.
Yep.
Okay. Yep, and also those labs who have a device, once they've finished the data collection, would be great if they could also then, offer to share their device. So the way that we've handled it, basically, I've asked the lab in China to handle all the shipping fees, and then someone from, I think, DHL, showed up, picked up the box, and they took care of the rest. So it was very easy for us to share the device. And also the lab that requested the device, I think it would be good practice if they took care of the insurance, so they make sure that there's insurance in place to cover the device if anything gets damaged. All right. We have 4 minutes for questions. Ines.
Not a question. So I was just more to say, remember to use the SOPs, share on Discord if you're collecting data, starting if you have any questions. And before uploading to OSF, please check the SOP and check that the data matches how the data should look, that has the data structure, okay? Because currently you're free to upload, but we want to minimize us checking if you're doing it properly. So if you please can check, as this will make things easier once we start data analysis.
Till?
Yeah, I also have another poll. So maybe if all those labs who are planning to use EmlaCream could raise their hands once so that we get an idea.
Okay, so that's at least
Yes.
5. Good. So that's 100 participants with EMLA. That's good. All right. So before we all leave, so this was our kick-off meeting, and I guess the name implies there will be more such meetings. So what do people think? Should we have more such meetings where we all come together? And what time frame would be good? I would initially assume that like a quarter yearly meeting might be good, like have such a meeting basically every 3 months. So we could, have one, what was now October. So we could have the next meeting early in the new year. Could you give me a thumbs up if you think that's a good idea? All right, getting a couple of thumbs up here. Okay, so then we'll be in touch with the next meeting for a date somewhere mid-January, I guess, when we all have recovered from our New Year's hangover. Then we can reconvene. All right, I would like to again express my deepest thanks to all of you. This call, I have to say, is quite overwhelming with so many people. It just makes me realize how many of you are supporting this initiative, and I'm deeply, deeply grateful for everyone who's supporting this. And I guess I speak on behalf of the whole steering committee. So thank you all very, very much. This is super, super exciting. And although it wasn't a big pleasure for me personally to enter 132 names into the manuscript portal of Nature that doesn't allow an automated upload, it is the pain absolutely paid off seeing all your faces today. So thank you very much. Bye-bye, and good luck with data collection. See you in the new year. Bye-bye.
Ciao, Simon.
Goodbye. Thank you.
Thank you.
Bye.
all.
Thank you.
Bye-bye.